So, good evening, everyone. And as they said, my name is Liliane Mukaramera, and I'm going to talk to you about Cryptococcus. So, Cryptococcus is a yeast, and these images represented here on the slide, the image that I took of Cryptococcus using the microscope. So I thought it would be good to introduce myself so you know who's talking to you. I was born and raised in Rwanda, and Rwanda is this tiny country. You can see in red on the African map. So we are located in Central East Africa, and that's where I did all my initial education until I finished my bachelor's degree in biology at the National University of Rwanda. Then I moved to Scotland, where I studied for my postgraduate studies. So both my master's and PhD was at the University of Aberdeen. And during my PhD, I worked on a fungal pathogen, another yeast called Candida albicans. This is the one that caused thrush. And I was looking how different forms are recognized by human immune cells. So those were the cells that were taken from the blood. After my PhD in 2014, I moved to Minnesota in the US, and I spent there 5 years studying Cryptococcus. And my project was looking again at the morphological chitin cells. So when cells become really big, and I was studying why they were changing and increasing the form. And then since 2019, I am an exeter. I'm a lecturer in the MRC Center for Medical Mycology. That's about me. And these were just the images that I thought I put here to show you how Rwanda looked like. So it's called also the land of 1,000 hills, where we have so many hills. And these were like, on this image one, you see the tea plantation. So we grow tea, that's one of our main exporters. And we also known to have volcanoes, and we have mountain gorillas in the volcanoes. So if people have saw the movie Gorilla in the Mist, that was in the volcano in Rwanda that was filmed. So coming back to my talk, I'm going to talk about Cryptococcus, a sugar-coated yeast. And I decided to divide this talk into 3 parts. The first one is I'm going to talk about what is Cryptococcus. And the second is why should we study? Why do we study Cryptococcus? And then the third is when I'm going to talk about its sugar coat. So first, Cryptococcus is a fungal pathogen. So it's in a kingdom of fungi. It's a basidiomycete, and there are more than 34 species of Cryptococcus, but only 2 species are found during human diseases. So the majority of diseases in human are caused by Cryptococcus neoformans and Cryptococcus chitin. So on your right, you can see there are images of Cryptococcus. Again, the top one have been taken using an electron microscope, and the bottom one is just the light microscope. And you can see that you see different things depending on how you look at it. Where do we find Cryptococcus? So Cryptococcus is ubiquitous, so it's everywhere in the environment. It has been isolated from the trees, from the dirt, bird guanos. But in addition to human, Cryptococcus can also infect rodents, reptile, bird, and soil microbes. So it's really almost everywhere, and we breathe it every day. On the left is a diagram representing what we believe how human got infection. So we believe that the infection starts when we inhale spores or desiccated yeast from the environment. And then this Cryptococcus will go into the lung. And then here, that's where the first time it meets some cells of immune system. Either it can be killed and eliminated, or if the immune system is weak, Cryptococcus can disseminate through different parts of the body and can reach the brain. And this is the case where people develop meningitis, and people that comes to the hospital with Cryptococcus infection, mainly they come at this stage where they have meningitis. And the Cryptococcus that we normally study in the lab are the ones that have been isolated from the cerebral spinal fluid of patient. So those are the cells that we use. So why study Cryptococcus? We study Cryptococcus because it affects many people. So it's estimated that every year there are around 1 million of infection. And it's also, in addition to this, it has a really high rate of mortality. So between 20 and 70% of the patient will die from Cryptococcus. Recently, the WHO have published a list of fungal priority pathogen, and Cryptococcus again comes at the top of the critical group. So these are the pathogens that are affecting many people, and that we still need to find out a lot about them that we don't understand, don't have enough drugs. So this is at the top of the critical group of the fungal priority list. So this is why we're studying Cryptococcus. So who is at risk of Cryptococcus? They are, as I mentioned, that there were 2 species, predominantly in human. Here, also when you see the people who are affected are divided kind of into 2 groups. Cryptococcus neoformans mainly affect people with weak immune system. So it's mainly people with advanced HIV/AIDS, people who are receiving organ transplant, but also people who are taking steroids, people who have cancer, diabetes, rheumatoid arthritis, or any other medication that weaken the immune system. So those are the people who are at risk for developing Cryptococcus neoformans infection. When we look at the Cryptococcus gatii, there is different characteristics in different parts of the world. For example, in Australia and New Zealand, people who had Cryptococcus gatii infection were people who were otherwise healthy. So there was no known underlying diseases like HIV or cancer, who are receiving cancer chemotherapy. So it were people who are healthy. They tend to be male and of Aboriginal origin. So those were the people who were coming up with Cryptococcus gatii infection. But when you look at the outbreak that happened in the British Columbia and Canada, there were people who have weak immune system, so similar to some of that were affected by neoformans or have other lung infection, and they tended to be people who are above 50 years old and who smoke tobacco. So combining this with the previous one, you can see that there is a long list of people who are at risk of developing infection caused by these 2 species. So in addition to that, Cryptococcus is associated with high mortality. So here, this diagram is only showing estimates from people who are living with HIV. So we're not counting other group. And with this, if you look, I decided to make this graph dividing into the part of the world where people are coming from. So if you look into lower income countries, you have around 70% of people who are dying in care. So in care, it means the people who are access to the hospitals and who have medication, and not in care in people who have not access to the hospital. And those low income countries, you can see that if you don't even have access to the hospital, you die, 100% of those people will die. You might say that, yes, it's understandable, there is a lack of resources, but the worrying part is when you look at the North America and Europe. So in this case, patients have the best available treatment. They have access to the hospital, they have the access to the treatment that we have, but you still have around 20 to 40 people dying. So this is, I think for me, we shouldn't accept that. It shows that what we have is not working and we need new drug. But also I will highlight that this estimate in people with HIV, we actually really don't know the burden of Cryptococcus in non-HIV patients. So it shows you how we still have a lack in this part, and we need to investigate and know the real burden of Cryptococcus infection in other group of patients. So combining this, I conclude that we need new and better antifungal drugs to treat this infection. In order to have new antifungal drugs, we need to understand how this pathogen causes disease. So this leads me to the third part of my presentation about the sugar coats of Cryptococcus. So why study the external surface of Cryptococcus? I call the sugar coat because Cryptococcus has two coat or envelope at the outside, and they are composed of carbohydrates. So the sugars, and that's where the name came from. So why should we study this? First, I like to use this analogy using the sweet, like if the surface structure can direct recognition and identification. So if you touch these different candies, you can differentiate. You can differentiate Skittles from M&M. And if you, like these images have different candies, but if you touch them by just the touch, you can know and identify. So the external surface and external structure can direct the identification. But it's not just the external structure. We need also to think about the composition. This image shows you M&Ms, but if you touch them, all of these M&Ms will feel the same. So to the touch, but they are different. They might be different. We know that the flavors of M&M are different. You can have milk chocolate, you can have mint, you can have peanut. By just touching them, you won't know the flavor. So to really do a in-depth identification, we need to know both the structure, but also the composition. So this is the same when we're thinking about fungal pathogen. Why do we need to know this? Is that these external components are the ones that are recognized by immune cells. So on this diagram, I'm showing you here, it's a Cryptococcus cell, and at the cell surface, these are different polysaccharides that you can find. Some of them are in the capsule and in the cell walls. Those are the two coats that I was talking about. And these are receptors. So they're like detectors that are on the surface of immune cells. So these receptors, we recognize different components. For example, a receptor called Toll-like-4 recognize the GXM from Cryptococcus. But if you look for the detector, this receptor called dectin-2, this one is going to recognize alpha-mannan. So using this, you see that if you change the cell surface, then you're going to change the recognition by immune cells. And if you change the recognition, this will result into a different type of immune response. So this is why, so change in the cell surface will affect how Cryptococcus is perceived by immune cells. That's why it's important to know what is at the cell surface, what is being interacting with human cells, and what is being recognized by cells of immune system. So Cryptococcus, it changed morphology in different environment. Here I'm showing you in vitro cells. Those are the cells grown in the laboratory on the top. And then at the bottom, we show the cells that we obtain from infection. These images show you that the surface is completely different. Here, these images have been taken using, so the first two have been used light microscope, and then the other were the electron microscope. But you can see that the surface is changing when we're talking about cell we grow in rich media in the lab versus the cells that are present during infection. So I think it's important to know why Cryptococcus is changing. Why is it switching its external surface component? So what's the difference between in vivo and in vitro cell? First, Cryptococcus has two coats that I was talking about. There is one called the capsule. This is the external one, the one when you see the red arrow, and is composed by mainly two polysaccharides. One is called gluconoxylomannan, or GXM for short, and then that one is composed around 90% of the capsule. We have gluconoxylomannogalactan, and that's around 80%, and then we have some mannoprotein. These mannoprotein, some people think they're part of the capsule, other people think they're part of the cell, and they are being excreted outside. But this is one of those sugar coats. The second part below, so underneath the capsule, is the cell wall. And I am particularly interested in the cell wall because most studies that have been done on Cryptococcus focused on the capsule. So we really still need to understand the cell wall. From what is known, we know that in the cell wall, we have different types of polysaccharide. We have beta-glucan, alpha-glucan, chitin, chitosan. We have also melanin and some protein. The problem with this, when I started working on this, I found that this component, or what we know, came from the cells that have been grown in the lab, in vitro, and the cells that did not have the capsule. So we really didn't know. And from the image that I showed you previously, you can see that in vitro and vivo are different. So I really wanted to investigate what's happening with in vivo cells. And I wanted to use wild type cells because if we use the mutant that don't have the capsule, those ones normally don't cause disease. So I think it's important to study a virulent cell that we know can cause disease so that we can correlate what we study with a disease and the virulence. These images shows you again, cell grown in the lab, cell that came from infection. Here it was an infected mouse, and Cryptococcus are those cells in red and green. So you can notice the difference in the size between the green and the red. And the gray cells are immune cells. And then the other image on the right is image taken on a sample from a human patient. And you can recognize again cells of different sizes, bigger and small. And this shows you that in vitro, we have cells that looks like the same, almost the similar size. But when we go in vivo, then we have these cells with varying sizes, and that's where we have these huge cells that have been called Titan cells. And they are present both in human and in rodent infection. So I wanted to study this and see what is the cell wall component, cell wall composition of these big cells, Titan, and the small cells, and in vitro grown cells. The image on the top shows you the cells that I used from the in vitro cells, in vivo typical. So these are the small one, and in vivo Titan cells. I want to mention that for the in vivo cells, I have to irradiate them. So I have to remove the capsule so I can study the cell wall. What you can see at the bottom is although I found similar sugar in this cell wall, the amount of sugars the proportion was changing. For example, you see that when you compare in vivo to in vitro cells, we have an increase in the chitin. That's seen significantly in glucosamine. This is the molecule that make chitin and chitosan. And you can see also that we have increased mannan in titin cell. So there is here show that we have difference in proportion between in vitro and in vivo, but also in vivo. These were the cells that came from the mouse. But when you take the small one and the big one, the cell wall is different. So this was one finding, that although we see cells during infection, all those cells don't have the same cell surface. So why? Why is Cryptococcus changing its cell surface composition? One of the things we looked at initially was the chitin, because chitin have been found to influence immune response in other fungal pathogen. So we looked at chitin. And here, the cells that I'm showing you, you see the images. When you see cells that are coming blue and green, so it's increased in chitin, more chitin, and the darker is less chitin. We used a wild type, but also we used two mutant. One mutant was called RIM101. It's smaller cells, but they have high chitin. And then another one, GPR4 and GPR5, were smaller and less chitin. So that one helped us just focusing on chitin rather than chitin and wondering if also the cell size is doing something. First, we looked when you infect the mouse with these three different strains, that you're having the similar number of cells in the lung. So this is showing that all those three strains were able to multiply similarly in the lung of the mouse. But when you looked at immune cells that have been produced in response to that infection, you see differences. You see the wild type and the RIM101, those were the one with high chitin, that there is an increase in the immune cells that called Th2. So type 2 immune cells were higher when you have high chitin. So, and this was irrelevant. The size did not matter, because you see the same thing with RIM101 that has high chitin but small cells. And we looked at the survival of the mouse, and you can see that when you have high chitin, so this is RIM101, and the wild type, that is K99, that the mice died earlier. And when you have less chitin, that the mice survived. So they died, but they died later. So we concluded from that increase in chitin content was associated with a non-protective immune response and worsening of the disease. So this is one proof showing a cell wall influencing disease outcome in the mouse. While we were working on that, there was another group in the US. For them, they looked at chitosan. That's another cell wall component. And they used mutant similarly, and they have here on the left diagram, you can see the CD1 mutant have less chitosan comparing to the other strain tested. And when you look at the survival of the mice infected, you see when you have less chitosan, around the 60 days, you still have 90% of the mice that have been infected. But when you looked at a strain that has high level of chitosan, all mice died before 20 days. So this was another proof that chitosan, so when you have a reduced chitosan, we have a reduced virulence, linking again Cryptococcus cell wall to the virulence of the strain. So cell wall chitin and chitosan are needed for Cryptococcus virulence. But the thing gets complicated in Cryptococcus because as we go on, research is showing that we actually have more morphology during infection. So initially, when I started my study, we had typical, we knew typical and titin cells. So there was a group that found that the other cells that I called drop cells, those were the cells that are really small comparing to the one above, but these were metabolically inactive. So that group that described them proposed that maybe this form is used during dormancy. And then later on, another group described the cell that they called titanide. Those were less, so because they believed that these cells were coming from the titin, but they were smaller. So titin are between 10 and 100 micron of cell diameter, so they are huge. But these titanide had less than 4 micron. But these were different from drop cells. They were metabolically active and they were oval in shape. While normal, a typical Cryptococcus is round. It's nice and round. You see that it's really, but you can see the titanide were oval. So we're still getting more morphology. These titanide have a thin cell wall, but we don't know what is in there. So that paper did not look into the sugar and see what is changing. This year, there is another morphology that just being described, and it have been called seed cell. They are smaller. They are similar in size and form to drop cells, but they are metabolically active. And they propose that these cells were the one that aids Cryptococcus in dissemination. So they think in the lung, as this diagram shows, you have bigger cells. This one in purple, you have typical cells, and then you have the smaller blue cells that they called seed. And these seed are the one that will disseminate in the blood and will pass through the blood-brain barrier and reach the brain. When they analyzed these cells, they found that they have high mannose exposure on the surface. And so this is again showing that when Cryptococcus change morphology, it changed the sugar at the surface. And they found that this mannose, the mannose exposure, mediate phagocytosis by macrophages. So recognition by immune cells called macrophage, and also engulfment by these cells. So combining these things, you see that during infection, Cryptococcus change morphology. By changing morphology, it changes also the cell surface, the sugar at the surface, and this helps Cryptococcus to cause disease. So chitin and chitosan were associated with non-protective immune response, and mannose exposure in the newly characterized seed cell is mediating phagocytosis by macrophage. So this image, that the ability from Cryptococcus to switch from this top image to the bottom is switch and changing sizes of the cell, changing the sugar at the cell surface in order to cause disease. So what about other cell wall components? So this is a question that is still there, and we're working on this to try to know, because we saw that everything is changing. Glucose is changing, mannan are changing, proteins, all those. Why is Cryptococcus changing? All those other cell wall components. So this is a question for the future that we don't know the answer right now. So what am I working on right now? I told you that I'm interested in the cell wall, and specifically the cell wall during infection. So first, after I worked on titin and typical cell wall, so those were images we took with electron microscope, and you can see that the cell surface is different. We proposed a diagram. So it's like, we know this component, the amount are changing, but we don't know the localization, the organization. We don't know. We're proposing, this is basing on other fungal pathogen, that there is chitin in the inner layer, we have melanin, and then we have like this inner layer, in the middle layer we think is mannan, and then at the cell surface we have the capsule that's composed by GXM and GXM gap. But we need to confirm this. So this is one of the projects I am working on to identify where each sugar is localized. If we change the content, do we also change the localization? Which one is being at the top, at the bottom, in the middle, and how does that influence the immune recognition? A second project I'm working on is how does the cell surface, those sugars change during antifungal treatment. The reason I thought about this is these are changing during infection, but patient will be treated. They will be given drugs. And we know that drugs can change fungal cell surface. This has been observed with other yeasts like Candida and Aspergillus, but we don't know what's happening in Cryptococcus. So I want to understand how does the cell surface change when that patient are taking antifungal drugs. And I have a grant from the Academy of Medical Sciences to look at this. A third project that I am involved in is how do cell wall changes affect, or are they related to Cryptococcus-related iris? So iris is an immune reconstitution inflammatory syndrome. So during Cryptococcus infection, a portion of patients, around 25%, develop this iris. Iris is where you have an exaggerated immune response. So patients with HIV normally have lower CD4. But when they are being treated by antiretroviral, then they recover CD4. But by recovering CD4, some patients will develop an exaggerated immune response. And actually, these people will die not from Cryptococcus, but from that exaggerated immune response. And this diagram that has been published show that when you have people who will have this iris syndrome, you have less chance of survival comparing to patients with a similar background who also have Cryptococcus but without iris. So iris is a major complication of Cryptococcus, and it's not really understood why some people will have iris, other people not. And for me, my hypothesis is that I think the strain or the pathogen that's infecting those people might be different. So I am currently analyzing the isolates that we got from patient with iris and those one from patient without iris to see if the cell wall and the cell surface is changing and if that's why those people develop iris. Because otherwise, you don't understand. They look the same, they have similar level of CD4, they are getting the same treatment, but some people develop iris, others not. So a take-home message. So even if you didn't understand anything that I said, take home this 4 points. So fungal pathogens are a serious threat to humans. Normally, when we think about microbes and infection, we think about bacteria, and we forget about the fungi. So fungal pathogens are also a serious threat to humans. One of those fungal pathogens is Cryptococcus, and Cryptococcus affect an increasing number of human patients. I say an increasing number because we know patients with cancer are increasing, people who are receiving organ transplant are increasing, people who are taking medication that weaken the immune system are increasing, and those are the people who are at least of Cryptococcus infection. So there is a huge problem of those people who are at risk of developing Cryptococcus. During infection, Cryptococcus change and exist under several morphological forms. And those forms have different cell surface. So they have different cell wall and capsule composition. And those cell surface changes are helping Cryptococcus to cause disease. So one of these that I studied, the cell wall, for me is important because human and mammalian, we don't have the cell wall. So if we can design a drug that will go and target the cell wall, then those drugs can kill the fungus without affecting human cells. So that's why I think the cell surface, the cell wall is really important. And if we can understand how it's being modulated during infection, then we can design a drug that targets that cell wall. And the drug will have a small side effect to human cells that don't have the cell wall. So I would like to end my presentation acknowledging people who helped me do this work. So at the University of Exeter, my mentors, my colleague, I have collaborated at the University of Minnesota, and then University of Cape Town and Makerere University in Uganda. That's where there's a clinical trial, and I went there to study patient, and I get samples from patients from Uganda. And the whole MRC Center for Medical Mycology at Exeter and the Academy of Medical Sciences for funding. And thank you for your attention.
Thank you for that. That was a wonderful presentation. I mean, I didn't get all of it, but the nice thing is we're recording it so I can look at it again. So, anybody has any questions? If you've put them in the chat, we'll try and address them now. And hopefully, Liliane will give us the answers.
Yeah, thank you. We've got a couple of questions come through, Liliane, already. But if you do have questions for Liliane, type them now and we'll read through them. So, first of all, a question from Angela. Angela says, it's a fascinating talk. Angela asks, why does Cryptococcus affect the human brain so much, whereas other fungi that cause fungal diseases like Candida and Tinea don't and just affect the skin and guts?
I really don't have an answer for that. But so it's it goes everywhere, but it kind of have like we prefer to go to the brain. So people will come with meningitis and they will suffer with meningitis, but when they started doing autopsy in Uganda, so they started doing autopsy from people who die from Cryptococcus, they found that Cryptococcus was actually everywhere in all other organs, but because patient came with meningitis and you have a lot of them in the CSF, so initially people were just thinking of the brain and the central nervous system. But if you look at all the other organs in the autopsy that have been done that I'm aware of, they found Cryptococcus in all the organs.
Okay.
Yeah.
Okay, that makes sense. Another question from Neil. Neil asks, Liliane, you explained that Cryptococcus can infect a wide range of animals and even amoebae, and that the wall is different when grown in the lab or when causing an infection. So how similar or different is the capsule on the wall when it's in a range of different hosts?
I don't think we have really thought that. We don't know anything about this. Initially, when I started, the actual information that was there was for the cell that would have been grown in the lab. And then for me, I went and used the one in the mouse. One that I really wanted to look at was from a human, and I really plan to go and get sample from human and immediately analyze them to see how is in the wall during human infection. The amoeba, they can eat Cryptococcus, and we think actually that was associated with why sometimes it was hard to get samples from the soil. So if you talk with people who have worked with Cryptococcus, you take the soil, you isolate like DNA, and then you see that there is Cryptococcus. And if you put your soil in the fridge, for example, and go like 3 months later, you test again, the sample is negative. So you can't see Cryptococcus. And the lab I was in when I was doing my postdoc, we struggled with that. And then at one point, someone said, we know amoeba can eat Cryptococcus. So maybe amoeba in the soil are eating Cryptococcus, and they proposed to store the soil at -80 when it's really cold. And when you store the soil at -80, the sample still, Cryptococcus still there even after 1 year. So I think amoeba might be eating Cryptococcus. I don't know if we can recover cells from amoeba. Maybe we can try, but I don't think if anyone have really looked at the cell wall. So for Cryptococcus, the cell wall is still an area of understudied. So we don't know. There's a lot that we don't know.
Okay.
Yeah.
Another question from Dimitro. Dimitro says, thank you for the talk and very interesting results. If I understand correctly, GXM capsule is located around the cell wall in clinically relevant cells. What do you think explains the difference in non-protective immune response and mice mortality you found with varying cell wall composition? And Dimitro suggests surely it would be the GXM composition that's relevant, as it is the outermost layer that the immune cells will encounter.
So when people make diagram, especially of the cell surface and the cell wall, so we have like model organism, and people normally propose depending on how they understand. And there is still a question that for me I know that we say that how chitin that it's believed to be underneath the capsule is recognized. The reason we know that it's chitin is that we took a commercial chitin. So we took those mutant, we actually added pure chitin that we bought from Sigma. And we replicated, we got the same type of immune response. We got the mice that died faster. So with that, we concluded that it was actually chitin because we replicated with using just pure chitin. I think that's why I said one of the projects I work on is to look at the localization. I think there's somehow, maybe we're wrong, that thinking there is layers after layers. Maybe it's just like one is somehow interacting, or there is a cross-linking, but they are exposed, or somehow they interact with the host cells. Because if you see the diagram in all the literature that we have, that people think there's different diagram, but most people think that the chitin is underneath, the chitosan is underneath, but those are the ones that actually is thinking that they are influencing the immune cells. It's like, how are they recognized? Is really the GXM covering the whole thing with no access, or do we have in the middle of the GXM some maybe chitin fibers and chitosan? So I think that's something we really need to understand, because for me, I believe that it's not just like GXM that's covering the whole thing and doesn't have access. Yeah.
One question from Reese. Reese says, are there lineage-specific differences in the cell wall?
That's something I really, really want to do. I started with one serotype, and the reason to choose that it was because it's the one that majority, it causes 90% of infection, but I know there are different lineages. And I really want to do that and to see different lineages, different species. Do we have the same cell wall? I think my guess will be not, because when you have those lineages, they don't cause the same type of infection. So but it's a guess, but it's something that I'm interested in doing and analyzing.
Reese also commented, it would be great to study further in amoeba. He agrees.
Yes.
And a fascinating point about Cryptococcus being found in other organs too.
Yes, it is, but I don't think people have, because it was a small study that looked at the autopsy, and I don't think anyone have also looked at other organ. Why is there? What is doing? Is this related to why patients are dying? Or
I had a question, and it's less informed than some of these questions, but it's about the cell morphologies. So you're identifying lots of different cell morphologies for Cryptococcus. Is that similar to other fungal pathogens, human fungal pathogens? Is there the same range and diversity of morphology?
I think it's different. So most fungal pathogen will have different form, but most we have a filamentous and the yeast. So you have round and filamentous. But in Cryptococcus, what we're noticing, you can induce filament in the lab, but there's very few cases where they were observed during infection. But the difference with Cryptococcus is the morphology that are being described, they are all yeast. So they are yeast, they are just different sizes. And they are only forming either during infection or when we use a host mimicking condition. So the difference between Cryptococcus with other fungal pathogen is that it's not just yeast hyphae, yeast filament is yeast, but yeast of different sizes.